We have been talking about biological structures which interact with drugs, and I want to focus again, as I said last time, from the viewpoint of treating the biological structures as organic molecular entities, because after all, that's really all they are. And we will see as we go through this that the kinds of things that we have talked about through 14C and 14D still apply here. Non-covalent interactions play a big deal. How reaction mechanisms occur, to the extent that we talk about that, those same kind of things apply, even though the nucleophile might be a bigger nucleophile. The leaving group might be a bigger leaving group in a biological molecule. It's still exactly the same kind of things that work. All right, so we've been talking about receptors. We'll talk more about enzymes today, but receptors are proteins that bond some sort of molecule, and when this molecule is bound, it signals some process to start. And in a generic sense, we can think of the receptor as having a specific shape and size for its active site, and so the signal molecule needs to fit that. The signal molecule needs to have the right shape, the right size, and also the same kinds of interactions that are this complementary kind of non-covalent interactions to offer to the receptor. So, for example, if the receptor is very polar, then the molecule that it docks needs to be very polar to be accepted. These issues of shape, size, and complementarity when it comes to non-covalent interactions are absolutely critical if you are trying to design a pharmaceutical that might target that particular receptor. Receptors come in different flavors, or the drugs that interact with receptors come in different flavors. We have antagonists and agonists. And the antagonists are drugs that bind the receptor. They usually have very strong affinity, but when they are bound, they have little or no intrinsic activity, meaning they don't cause the receptor to fire. So these are things that block the receptor. On the other hand, there are times when we need a drug to go into the receptor and make it fire. That may happen, let's say, if there's a deficiency of the normal signal molecule. And in those cases, we call these drugs agonists. These are things that have a strong affinity for the receptor and trigger the receptor, cause it to send out its signal. Sometimes, many times, agonists actually have stronger affinity for the receptor, stronger binding than the natural substrate. Some examples that we talked about: curare, which interacts with neurotransmitter receptors and can paralyze breathing, for example. Famotidine, also known as Pepsid, is a gastric acid production blocker, and it interferes actually with a receptor called H2. It's a histamine receptor, the same stuff that gives you allergies. And histidine is the signal molecule which says to the gastric acid receptors, hey, food's coming in, make more gastric acid. Or an exam is coming, you're upset, make more gastric acid. Right? You probably are becoming more familiar with on a daily basis that particular process. And so famotidine is a drug which actually blocks that receptor but doesn't cause it to fire, so that would make it an antagonist. Prozac is one of a group of serotonin uptake inhibitors. What it does is it sits in the serotonin receptor and blocks it from new serotonin coming in, which helps modulate neurotransmitting and mood and the things related to that. All right, now let's switch to enzymes. Enzymes have many similarities with receptors, but there's some important differences. Enzymes are biological molecules, most of the time proteins, but sometimes other things, that carry out chemical transformations. That's different from a receptor. When a receptor blocks a signal, when a receptor docks with a signal molecule, they say hi to each other, and then when the end, the signal molecule is released. An enzyme, on the other hand, carries out chemical reactions on the molecule that it binds to. Maybe one mechanism step, maybe multiple mechanism steps, but it carries out this reaction. So instead of calling it the thing that's docked the signal molecule, we call it the substrate. Now, of course, we've talked a little bit before about enzyme catalysis and how it works. There's very large numbers for catalysis. 10 to the 28th is the record, but 10 to the 9th, 10 to the 12th kind of acceleration relative to non-catalyzed reactions is pretty common. And just to remind you of some of the things we've talked about, the ways that enzymes catalyze things, one of the things they can do is hold the reactants in close proximity and with the correct orientation for the reaction. Sometimes this is called the chaperone effect. And what that does is basically it gets the molecules to line up so that for all intents and purposes, every collision between the reactants is productive. Remember that for reactants to react, when they collide, they have to collide with the appropriate orientation so that the right orbitals are lined up. Another common thing that enzymes do to catalyze reactions is stabilize the transition state. This was Linus Pauling's idea. By stabilizing the transition state, you can bring the energy of activation down. Or you can destabilize the reactants and or do both at the same time. But anything you do to bring up the energy of the reactants and or bring down the energy of the transition state is going to make that faster. Now, we talked about this a little bit when we talked about chymotrypsin hydrolysis a while ago. You may find it useful to review that. Now, to think about how enzymes work, we have a little cartoon here in the middle, which is somewhat similar to our receptor cartoon, except here's our enzyme, here's our substrate. Notice the complementarity in the shape and the size. So here the substrate has a little square thing sticking out on it, and the enzyme has a little square hole, so they fit together nicely. The shape, the size, the polarity all have to match. But here's where a difference is. At this point, once the receptor has bound its signal molecule, it sends off a signal, and the signal molecule is released. But an enzyme grabs onto that substrate, does chemistry on it, changes its structure, its chemical structure in some way. Oxidizes, reduces, hydrolyzes, does all sorts of kinds of things that enzymes do. But in the process of doing that, most of the time it's going to change the shape of the substrate. Now here I have it going from square to triangle. There is no enzyme whose job is to convert squares to triangles. All right, this is just an illustration of what we're talking about. But because the modified substrate now doesn't fit as nicely as the substrate to begin with, it's not bound as strongly, and it can be released. That's important because the job of an enzyme is to be a catalyst, and a catalyst has to be turned over. So you've got to free the modified substrate so that the enzyme catalyst can go back and react on another molecule. And so the kinetics of this process obviously are going to be a little bit more complicated because there's more steps in here. Now, what do drugs do with enzymes? Most of the time, what we do with an enzyme is we inhibit it. It's kind of difficult to make it go faster, to do its job more frequently. There are some enzymes that work that way. There are some drugs that target enzymes and make the enzyme work faster. Those aren't as common. Most of the ones that we deal with, though, are inhibition. And by the way, things that act on enzymes, this is the most common pharmaceutical category that's out there. So what kinds of things? Well, we're inhibiting enzymes. Of course, we want to block its action in some way. Now, we can block its action temporarily, or we can block its action permanently. If the inhibitor binds in a non-covalent or reversible sense, then that can be released. Maybe or maybe not it's acted on by the enzyme, but it can be released. On the other hand, if it binds covalently, forms a covalent bond to the enzyme, chances are that that's the end of the enzyme. The inhibitor is now permanently sitting there in the active site. It can't be moved out. Imagine, if you will, as a metaphor, let's see if this works. This is your parking space. Okay, somebody else comes along and parks their car in your parking space. You are now inhibited from parking your car in your parking space. But maybe later on they back out and you have your parking space back. That is non-covalent, reversible inhibition of the enzyme which is called your parking space. On the other hand, if a person crashes their car into your parking space and they kind of drive it up on the post that's at the end of the parking space and they just abandon it, so there's this piece of junk car now sitting in your parking space that isn't going to drive away. That's kind of permanently blocked. So that would be the covalent bonding kind of process. If that doesn't work for you, just forget the metaphor. All right, now, so the thing that becomes permanently bound we call a suicide inhibitor because the molecule kind of commits suicide in the process of binding. That doesn't, it does not mean this is a drug you take that prevents you from committing suicide. That's not what we mean by this. Oh, oddly enough, there are some suicide inhibitors that actually are suicide inhibitors in that way. If you're depressed and you need a drug that does that. They're called suicide inhibitors because you can think of them as kamikazes, right? Remember kamikazes, airplanes crashing into ships, right? You're blowing up when you're crashing into the ship, you're committing suicide. But hopefully you're inhibiting what the ship is doing. So you're sacrificing yourself to cause something else to not function. So in this case, the drug, its structure is sacrificed. It forms a covalent bond with the enzyme, and that's the blocking of it. Now, another kind of inhibitor that's important are transition state analogs. These may or may not be suicide inhibitors. But remember that it's one of the ways in which an enzyme can catalyze a reaction, make it go faster, is by stabilizing the transition state. And so that means the enzyme has to kind of like the transition state. The structure of the active site has to be, it has to fit what the transition state would look like: shape, polarity, non-covalent interactions. Now, if you want to design an inhibitor for the transition, for the enzyme, you can't say, oh, I'm going to simply inject the transition state. You're not going to take a pill called transition state. Why? How long do transition states last? Not very long. Not a very effective drug if it only lasts one bond vibration. You know, by the time you went to the grocery store or the drugstore or the guy in the corner that sells you drugs, wherever you get your drugs from, it would be gone. Wouldn't be very effective. But you can have a molecule, a permanent molecule, not some transition state structure, but a real stable molecule whose structure looks an awful lot like the transition state. Its shape, its size, its polarity, where its charges are, looks a lot like the transition state. And therefore, the enzyme can be fooled into accepting it. It can say, hey, you look like the transition state that I'm supposed to stabilize. Come on in. And by that point, the enzyme goes, ooh, I really like you. You look a lot like my transition state, so I'm going to bind to you strongly. But I can't do chemistry on you, and you're not going to leave. So now I'm stuck. So that's what a transition state analog does. It kind of fools you. It's like if you, actually a good metaphor for this might be, anybody ever see the movie The Cable Guy? Right? So think of that as a transition state analog. It's a person, oh, it's the cable guy. He's coming to fix my TV. But once you get him inside, you can't get rid of him. All right. Here are a couple of examples of drugs that target enzymes. Actually, two we're going to talk about if we have time. One, sulfa drugs. These are among the first drugs that were designed to be pharmaceutical, designed to have a particular effect, manufactured on a huge scale around World War II or before then. And these guys inhibit an enzyme necessary for folic acid synthesis. Now, they turned out to be pretty useful because, as we'll see later when we get to talking about them, because bacteria synthesize folic acid, mammals don't. So if you mess with the folic acid pathway, human beings, mammals, we don't care too much. But bacteria have a real issue with that. Then there are penicillins, which are antibiotics that inhibit an enzyme necessary for bacterial wall synthesis. And unlike the sulfa drugs, which are temporary inhibitors, they're reversible, the penicillins generally work by irreversibly inhibiting that enzyme that's necessary for bacterial cell wall synthesis. So they are suicide inhibitors in that way. All right. Now, there are some other biological structures that drugs can interact with that are, currently there are less drugs on the market that interact with these, but at least in the case of interacting with DNA, this is becoming a bigger area. It's more drugs are being developed in this area. Now, if you're going to interact with DNA, there's a lot of things you can do. But remember that DNA's job is to store information and to be replicated when you're making the new cell. So anything you can do to interfere with that storage of information or the replication of DNA can potentially be a pharmaceutical target. This is one of the reasons why DNA is a popular drug target, is because there's so many things you can do to it, especially if you're talking about anti-cancer and that kind of thing. But it has a problem as an area for a drug target because every living organism is going to do this. So it means you have to be careful if you're trying to, let's say, kill a bacterium who's at the point where it's reproducing, and you want to target the DNA replication. You've got to be careful because the host will also be having DNA replication at the same time, and you don't want to mess with that too much. All right, so how can this happen? Well, one of the ways that you can interact with DNA is through a process called intercalation. And this is a non-covalent insertion of the drug molecule between DNA base pairs. Imagine taking your think book or your notebook or whatever it is you're taking notes in and sticking a piece of paper in it. Can I borrow your think book for just a second? Thank you. I'll give it back. So imagine this is DNA. I mean, I know it looks kind of like this, but right, DNA is a whole bunch of base pairs parallel to each other. there's your DNA. It's like a stack of papers. The ability for DNA to store information and replicate and so forth is a function of having that shape. If a molecule comes along and gets stuck in there, what does it do? It changes the shape of the stack. It causes distortions and bulging, etc., and that leads, thank you, that leads to difficulties in transmitting information and replication and so forth. So that's essentially what intercalators do. They get in there between the base pairs and mess things up. All right, so to be an intercalator, to slide in between the base pairs, to interrupt the aromatic stacking of adjacent base pairs, for DNA to say, hey, molecule, come on in, we want you to be here. You've got to offer something to the base pairs that interrupts the attraction between the two base pairs. It also needs to be of the right shape. And so the two things you really need to be an intercalator is, number one, you have to be planar, because you have to fit in between other flat regions. I mean, you can't, in the think book metaphor here I stuck my marker in, and that's kind of big, but it's much easier to stick in a piece of paper. Putting a watermelon in there would have been even harder. The closer the shape is to planar, better chance it has of going in. Number two, because the base pairs have all that aromaticity, they're interested in interacting with other things that are aromatic stacking. And so if the thing that's going to intercalate to be accepted should also be aromatic. So in other words, for DNA intercalators, we're looking to identify molecules which have planar aromatic regions. Now you might say the two of those go together. Of course, right? Aromaticity requires planarity. But just because it's planar doesn't necessarily mean it's aromatic. All right, so what kinds of things can happen from intercalation? Well, by messing with DNA, you can cause mutations. In other words, the molecule is a mutagen. That means DNA mutations are caused. These may or may not be repairable. They may or may not cause a problem with the biology down the road, but at least you've changed the DNA structure somewhat. Then there are carcinogens, which most of the time are mutagens, but carcinogens are things which, of course, cause cancer. And then there are mutagens which lead to fetal abnormalities, and these are teratogens. A good example of that, anybody remember a teratogen, a very famous one that we talked about in Chem 14C? Thalidomide, that is a teratogen. All right, so here's an example of a molecule that you may be familiar with. That actually is a DNA intercalator, and that is ethidium bromide. That's this molecule. Anybody hear of that thing? Yeah. This is why, when you're using ethidium bromide, you should be careful. All right, so here's its structure. It is flat, and it's aromatic. So 1, 2, 3, those three rings are definitely flat. They're aromatic. This phenyl out here is aromatic as well, but this is the main area that we're interested in. We use it for, or you use it for, a stain for nucleic acids in gel electrophoresis. So as I recall, you put the stuff in, or you run your gel up, you put the stuff in, and then you look at it with UV light to see where the fluorescence is, right? So this is what you're looking at, but it only is on the gel in the places where there are bits of DNA that it has intercalated into. And the problem is, because it can intercalate not only into the DNA that's in your gel, but also into your own personal DNA, which happens to be in your fingers, elsewhere in your body, and so forth. I mean, if you, God forbid, you eat this stuff, then it can mess with your DNA. So essentially, here's what happens. Here's we have normal DNA. It's our nice, beautiful double helix. And when the ethidium bromide intercalates, so now it's between base pairs. Here we've stuck it in between there's a GC base, or CG base pair, and there's an AT pair. So what it's done is it's caused a bulge in this region, because the DNA the backbone kind of has to move out to accommodate it, and because it's moved the bases far enough away, they're not hydrogen bonding anymore. So this area is now distorted, and if that particular piece of DNA is important for, let's say, coding for some protein synthesis or something like that down the road, well, you've messed with that, and so you can't make that protein, or maybe you make the protein wrong, or you turn something on which starts cell proliferation, uncontrolled cell proliferation. There's a whole lot of kinds of things can happen. But it happens basically because the DNA, the ethidium bromide, the intercalator, just slides in there between the base pairs. Now, there are some other intercalators we have on the top of the next page. The first example we have up here is andreamycin, which is an antibiotic. There are 1, 2, 3, 4, well, 3 flat rings that are aromatic, and the carbohydrate-looking portion out here, it's not a whole carbohydrate. It doesn't have all the OHs on it. But this actually probably serves to help it go to a particular place in DNA. So as this thing is moving down the chain, down the double helix, deciding where to go in, this thing is interacting with the phosphates and so forth on the DNA, on the backbone, and at some point the DNA says, hey, you're a molecule that belongs here. I recognize your carbohydrate portion. Come on in. Then there is quinine, which is an anti-malarial, the thing that works against malaria, and there is the aromatic portion. What all this other spinach does, I'm not exactly sure. I don't know if anybody knows, to tell you the truth. But there is the aromatic portion that is an intercalator. All right, now there's another group of drugs which is not as pharmaceutically, not as clinically important as they were hoped to be, but they have a very interesting mode of action, so I'd like to talk about them. And these are DNA cutters. And what they do is they damage DNA not by simply causing bulges in it, but by breaking covalent bonds. It's like taking your DNA and cutting it with scissors. So here is a prime example of a group, one of those drugs. It's called calicheamycin. It's a, shall we say, complicated molecule. Now, most of this stuff out here only has to do with how it recognizes DNA. As a matter of fact, bear in mind that a lot of these kinds of molecules, like quinine and calicheamycin, were not designed by Mother Nature to be DNA intercalators. They happen to, have any particular pharmaceutical property. Quinine is a product of a plant. Calicheamycin is a product of a fungus, as I recall. And so the fungus doesn't say, hey, I'm going to produce this product which is going to kill cancer cells in mammals. It doesn't do that. It's not quite that bright or that philanthropic. It happens to be an accident that it works that way. So a lot of times these kinds of molecules will have stuff hanging off them that really has no drug function whatsoever. And so if we're going to take a natural product, a naturally occurring molecule, and try to make it into a pharmaceutical, often we try to trim down the structure and see what is or is not necessary. It turns out in the calicheamycin case, you really don't need most of this stuff. It's out here. It is this portion of the molecule, the ene diene, the alkene with the 2 alkynes. That's the business end. So how does it work? This is how it works. It starts with this portion. We're just going to have the rest of the molecule. So the wavy line here means rest of the molecule, not important for our thing. So we start with this 2 sulfurs, this disulfide. That reacts with a nucleophile, usually an NH2 on a guanine in DNA. And the sulfur-sulfur bond can be broken. Now, this mechanism, by the way, is not complete. I've left out proton transfers and some other things. You can add those to complete it if you like. So that gives us now an SH, a thiol, which also is a nucleophile, and turns around and adds to that alkene. Now you're going to go, wait a minute. Alkenes are nucleophiles. They're not electrophiles, but alkenes can be nucleophiles. Alkenes can be electrophiles, as long as they have a way to accept electrons. So if you put a whole bunch of electron withdrawing groups on that alkene, then you can make it electrophilic. In this particular case, if you draw the curved arrows, you'll notice we're shifting the double bond now up here next to where the carbonyl is, and the carbonyl pi bond goes away. What we get is an enolate, and so that's why this particular alkene is susceptible to nucleophilic attack, because the product of that's an enolate. You can stabilize the negative charge through resonance. Now, in doing so, and again, I've left out some proton transfers and so forth, it looks like we really haven't achieved anything, but actually what we've done at this point is made a subtle change in the geometry of the ene diene portion. This reaction going from here to here takes the 2 ends of the triple bonds and brings them a little bit closer together. And when they come close enough together, the electrons move around in a reaction called the Bergman cyclization, named after Bergman at Caltech, who discovered this reaction actually in the lab before we knew that Mother Nature discovered it, long before we did. But it brings these 2 together, and it makes a benzene ring with 2 radicals on it. Now, what's the driving force for that? Making a benzene ring, right? It goes from being just conjugated to being aromatic. But in the process of doing that, this benzene ring that it's building has radicals on the opposite ends of the rings from each other. Bear in mind that this process occurs in DNA. So the DNA attacks the disulfide linkage that gets this whole thing started. This is the equivalent of holding a firecracker in your hand and lighting it. You're going, oh, look, firecracker. Shh, isn't that pretty? And what happens when the fuse burns down? Boom. You lose fingers, right? You take an eye out, that kind of thing. In other words, you go, hey, I've got this bad thing in my hand, and it hurts you because it's in your hand. So what you're doing is generating a diradical right in the presence of DNA, right next to the DNA. Well, what do radicals do? Add to pi bonds, hydrogen atom abstraction, all that kind of stuff. So the net effect is this diradical does bad things to DNA, and ultimately it gives you some DNA diradical, takes hydrogens away, which ultimately reacts with O2 and causes a cleavage of the DNA, so completely chopped across the double helix. And then there's the product of that. Now, this is actually kind of a cool mode of action, and because it's completely brand new. When people discovered this drug and how it worked, they were really excited because it was an absolutely brand new way for pharmaceuticals to work. And the advantage to that is, if you have a new pathway for pharmaceuticals that you haven't seen before, that potentially means that bacteria won't know it's coming and they won't have resistance. So these guys were really interesting. Or cancer cells won't know it's coming, they won't have developed a resistance to it yet. And as it turned out, these molecules were really biologically active. You only had to use tiny amounts, really tiny amounts to do damage to DNA. And that's also exciting because drugs that have lower amounts of material that you need to actually have a drug effect tend to be less toxic. Right? You generally don't want to take a drug where you got to take 5 grams of it at a time. That's usually not a good thing. But if you only have to take, a nanogram, that would be a great thing. But it turned out that this thing wasn't very selective. So it just chopped every DNA everywhere. It didn't care. Healthy cells, cancerous cells, it went after everything. So unfortunately, too toxic to be useful. But because of its mode of action, people really studied this thing. Can we make a simpler drug that's not toxic, et cetera? And the answer was, no, we can't, unfortunately. And so research in this area has kind of dropped off. But it was exciting for a while. All right, another class of drugs that mess with DNA that are actually still important are DNA alkylators. We saw on the last slide that guanine NH2 is nucleophilic. And you need that guanine NH2 to do hydrogen bonding. So if you mess with that guanine NH2, you can mess with hydrogen bonding. And the other thing you can do is if you form a permanent, irreversible covalent bond between that guanine NH2 and something else in the DNA, you can prevent the DNA from coming apart completely for replication purposes. And that's one of the ways in which some DNA alkylators work. So these are molecules that, form covalent bonds to nucleophiles, usually that guanine NH2, by doing some sort of alkylation. Usually it's an SN2 kind of process. And so again, they can interfere with base pairing and hydrogen bonding and replication and so forth. So one of the older ones that actually came out of mustard gases that were produced in even World War I was this molecule, mechlorethamine. That's this guy. Very simple. It's amine with 2 chlorines on it. And what does it do? Well, it starts by taking the nitrogen lone pair and in an SN2 process displacing the chloride. Now that gives us a 3-membered ring with a positive charge on the nitrogen. And you might say, why are we doing that? It builds ring strain. That's true, but it's also intramolecular. The lone pair is right there. The chlorine really can't get far away from it. So that gives us this 3-membered ring with the nitrogen and the positive charge, which is an okay leaving group. And a combination of that plus the ring strain means this is an electrophile. It is subject to SN2 kinds of reactions, backside attack. So if we take our guanine NH2 nucleophile, attack the carbon-nitrogen bond, open it up, that's pretty easy to do. It gets rid of ring strain, and it quenches the positive charge on that nitrogen. So now we have made a covalent bond to that guanine nitrogen. And again, I've left out the proton transfers. You're welcome to draw those in. So now we're going to do the same thing on the other end, make that 3-membered ring again, and take a guanine now on the other strand of DNA, close proximity, but on the other strand, do the same thing and build another covalent bond. Now what I've done is taken those 2 guanines, which normally may have only been associated only through non-covalent interactions with the other half of the helix, with hydrogen bonding. Now I have attached them with handcuffs, if you will. Right? Think about it this way. Let's see if this metaphor works. Imagine your fingers are hydrogen bonds. Right? DNA together, double helix, take it apart, replicate it. Pretty easy, right? Now imagine you have handcuffs. I don't happen to have any handcuffs with me. But imagine you have handcuffs. Okay, you can take your fingers apart, but your hands are still stuck together. You don't take apart handcuffs when you're doing that DNA replication. The handcuffs are like covalent bond. And so when you come down to take the DNA apart to replicate it, you get to that covalent bond, can't replicate the DNA. So then the cell is told, I'm sorry, you're not good enough to reproduce, you must go off and die. You know about that, right? Apoptosis, cells being told, I'm sorry, you're not good, you go off and die. That's kind of what happens here. It can be repaired to a certain extent, but for the most part, it's kind of deadly. Now, this process is not necessarily selective only for cancerous cells. This is generally an anti-cancer kind of treatment. Healthy cells have guanine, NH2 in their DNA as well. But the advantage to this is because this can be repaired, cells that are reproducing more quickly have less chance to have their DNA repaired before the DNA is tested to say whether or not it's allowed to reproduce. Because the cancerous cells reproduce much more quickly. So there's a lesser chance that the cancerous cell that's damaged this way gets to repair itself. So there's a greater chance that the cancerous cell is told to go off and die. But eventually you do build up enough damage to the DNA in healthy cells that, well, the drug can become toxic. This is part of the reason why a lot of chemotherapies, actually your hair falls out and you feel sick in other ways, just because some of that toxicity is building up. All right, let's talk about interaction with another biological class of biological structures, and that's lipids. These are generally less important, but there are still some drugs that operate in this area. Remember that lipids, the role of a cell membrane, which is built of lipids, is to keep the outer world out and the inner world in when it comes to cells. One of the things they do that's pretty important is maintain the sodium-potassium cation balance. So there's a certain amount of sodium and potassium cations that need to be inside, certain ratios, certain concentrations. If you mess with that concentration, then the cell can burst or die in some way. So what the phospholipid bilayer does, remember the core of that is nonpolar. It's all those lipid tails, nonpolar hydrocarbons. That's a no man's land as far as, or a no cations land as far as that's concerned, because the cations require polarity. Na plus and K plus, if you want to move them around because they're positively charged, you need polar structures to do that. The interior of the cell membrane is completely nonpolar, so the cations don't want to go there. So the cations that are outside stay outside, the cations that are inside stay inside. Imagine, if you will, here's another metaphor. We have lots of metaphors here. Speaking of cations and places they don't want to go, imagine, how many of you know that cats just generally don't like water, right? Some cats are weird, they like water. Most cats don't like water. So imagine you've got a house, and you've got cats inside that you love them, you want to feed them and keep them inside, etc. And there are feral cats outside, bad, flea-bitten, rabid, nasty, matted hair, stinky, and dumb names, right? Your cats have very nice names, Fluffy, that kind of thing. The cats outside are named Bill or something, I don't know. But they're just nasty names. You don't want them to come in. So what do? In your house, you basically put a big pan of water between the outside and the inside. The cats will come up and look at that pan of water and go, oh, water, that's pretty. I don't want to touch it. So the indoor cats to go outdoor would have to walk through the pan of water, and they go, I don't want to do that. The outdoor cats would have to walk through the pan of water, and as much as they might need it to take a bath, they go, I don't want to do that. So that region, which is anathema to the cats, because they don't like water, keeps the outdoor cats out and the indoor cats in. So the cell membrane, the nonpolar region in the middle of the cell membrane, serves exactly the same function as that pan of water. The cations on the outside stay on the outside. That's why that metaphor. The cations on the inside of the cell membrane stay in because they can't cross that nonpolar area. Now, cations and anions still have to move in and out as normal biological processes. And that often is done by the cell having some sort of molecular structure which chaperones the cations in and out of the cell. And such things are called ionophores, which literally means ion bearers. Now, they come in some different forms. There are molecules that physically move across the membrane. There are also ionophores that create what's called an ion channel, which is essentially a tunnel through the cell membrane, but a tunnel which is favorable to the cations and anions moving across. Think about it as, since I just see if this metaphor works. Down in San Diego, actually even further south, down towards the border, right, there's this big fence, in principle, that is supposed to be, you don't go across the fence, right? So you can't go from Mexico to the United States easily, and in principle you're not supposed to be able to go from the United States to Mexico easily unless you go through where the checkpoint is. That's the equivalent of the ion channel. It's a place where people are allowed to go. There's also tunnels that go underneath, which are the same thing. They're bad tunnels, but nonetheless, you can't go across the border, across the fence. You have to go through the tunnel. All right, so how does that work in terms of drugs? Well, there are some drugs that create either ion channels or transport, if you will, that goes through the membrane. And here's a structure of what an ion channel looks like. It's kind of a tunnel that the cations can move through. It's an area that's away from the nonpolar side chain, so it's kind of conducive to the cations moving through there. And one molecule which does a good job of building these things is called amphotericin B. That's this guy. And you'll notice that it has polar regions, lots of OHs, two carbohydrate-looking molecules, and a non-polar region where there is 1, 2, 3, 4, 5, 6, 7 conjugated alkenes. Now, the conjugation of the alkenes, I think, has nothing to do with the way this thing works. That's kind of just an accident. Again, this is a natural product that some other organism makes. I think fungi make this molecule. But the way this works is these guys line up to build the tunnel. The nonpolar hydrophobic region puts itself next to the nonpolar side chains in the phospholipid bilayer, and the polar areas, especially all of these OHs, tend to point in towards the middle of the tunnel. As a matter of fact, we have that picture on the next page. Tend to point in towards the middle of the tunnel. And all those lone pairs on all those oxygens make it conducive for the cations to move there. The carbohydrates that I've abbreviated here as carbo that sit on the end probably serve a cellular recognition role. In other words, because the surface of the cell membrane is studded with so many things, actually a lot of the cell membrane often will recognize a molecule that's supposed to be there by its carbohydrate groups. It kind of feels and says, do you have the right combination of things? Yes, you have the right carbohydrates. You're allowed to come in and be part of the cell membrane. So that's what having these carbohydrates are in there. That's how blood typing works, by the way. A significant difference between the blood types are the carbohydrates that's present on their cell surfaces. So this creates this channel, which got all the OHs in the middle, and that's a place that ions can move now in and out freely. They're kind of, the ions, the cations are kind of handed from OH group to OH group to OH group as they move down there. So the reason this will kill a cell is because it can allow a change in the sodium-potassium balance, and when you disrupt that, the cells can burst. Now, we've talked about some biological structures. Let's talk a little bit about how structure of the drug influences its binding. What kinds of things do we need to have? Well, we want to talk here a little bit about specificity. When you're targeting, let's say, an enzyme, let's suppose you're targeting chymotrypsin. Let us suppose for some reason you have a disease in which you have too much chymotrypsin. So every time a protein comes in, it gets chopped up, which is a problem because there are some proteins you need to have. So if you want to block chymotrypsin, you want to find a drug that is specific for chymotrypsin. You don't want it to block just any old enzyme, because what about the other enzymes you need to live? You don't want to just block every enzyme at random. Don't need to do that. So you need your drug to be specific. How does that come? Well, the specificity comes, again, as we've said before, by having the right shape, the right size, the right nature of non-covalent interactions. And the better the fit of the shape and the size and the non-covalent interactions, the better the binding is likely to be. And generally, the more specific your drug is going to be. By specificity here, meaning it's not going to target every single enzyme out there. You want to design a drug which is just going to go after, let's say, chymotrypsin or some other process. All right, so the binding itself can occur because of both non-covalent and, to a lesser extent, covalent forces. So the non-covalent forces are the same kind of non-covalent forces that we talked about in 14C, we used in the beginning of this course when we talked about solvent effects and so forth. The ones that are probably the most important ones are hydrogen bonding and dipole-dipole interactions, but there are ionic interactions. There's London forces can be an issue, etc. But hydrogen bonding is often a really big deal here. Then, of course, there's covalent bonding, which is the irreversible binding, and that's going to be a whole nother thing. You have to ask about if you're doing, let's say, nucleophilic substitution at a carbonyl, and that's the way the enzyme becomes covalently bound. Do you target the right position in the enzyme? Is it the right kind of functional group? So on and so forth. Now, there are other things that matter, however. For example, size. If the chymotrypsin active site that you want to block is a certain size, let's suppose it is this big. A drug molecule which is this big doesn't fit. So this drug doesn't inhibit this enzyme. No matter how pretty this is, no matter how much money you've spent developing this drug. By the way, the industry average is about $800 million to bring a drug to market, if you really like to know. So if you spend $800 million, doesn't work. It needs to be the right size. Now, what else does it need to be? It needs to be the right shape as well. All right? So our drug now is kind of spherical. Imagine your active site is kind of flat. Well, it fits in terms of shape, it fits in terms of size, although this is a little small. So the enzyme might say, wait a minute, that's too small. That's not my substrate. Don't dock. But if this is kind of flat and this is kind of round, it may not fit very well either. Another issue, very important one, is the stereochemistry. Enzymes are built from amino acids, which, with the exception of glycine, are all chiral. So that means if I have a substance built of chiral building blocks, most of the time that thing I built is also chiral. There are symmetry ways to make it achiral, but for the most part it's going to be chiral. So it's going to be handed. So in other words, I can have the left-handed version of the receptor, which may be the natural one, and the right-handed version one, which is the mirror image. So when my drug comes along with its three-dimensional shape, it may only fit one and not the other. A better way to say that is, since we don't have left and right-handed receptors, is if the receptor has the enzyme has a certain shape, when I make the drug, if it only has one stereocenter, that it can be either one of the enantiomers, but only one enantiomer is going to fit that particular receptor well. The other enantiomer may be ignored. Maybe it doesn't bind to anything and it just gets flushed away. Or maybe it binds to some other receptor and does something bad. For example, here's a great example of that. Thalidomide, molecule we talked about before. Thalidomide has a single stereocenter right there at that carbon. So it has two enantiomers. And as we said before when we were talking about stereochemistry back in 14C, those two enantiomers have different biological properties. The R enantiomer has the desired sedative. This was a drug that was given to, as they said in the late 1950s medical parlance, pregnant women who were hysterical. It doesn't mean they were running down the street naked screaming. That's not what we mean. It just meant that they were moody. So hysterical is what we said then. You imagine how un-PC that would be today. Right? Can you imagine the commercial on TV? Ladies, are you pregnant and hysterical? Take our drug. Now it's, are you pregnant and just not feeling right? Or there's other words that the FCC won't let you say on TV that are probably applicable as well. But so the desired effect comes from the R enantiomer. An undesired effect, a teratogenic effect, comes from the S enantiomer. So this means when you administer that drug, you need to make sure that the stereochemistry isn't going to be an issue. Now, when thalidomide was manufactured, it turns out it was easy to manufacture it as a racemic mixture. That's the way it was sold. So there was a mixture of both the teratogenic and the sedative effects. Now, in a modern sense, There are many drugs that have one or more stereocenters, and those different stereoisomers can have different properties. Sometimes there's one enantiomer, one diastereomer, which has the right properties, and all the other ones don't do anything. Sometimes the other stereoisomers do bad things. And then you need to make sure that your drug, that's when you manufacture it, that you don't have the other bad stereoisomers in there. Sometimes, there are a few examples of drugs that do this, the racemic mixture, or the mixture of stereoisomers, actually has greater drug potency than one individual enantiomer. That happens occasionally. Now, let me just finish this page before we quit today. So, exploration of the structure of a drug in relationship to its pharmacological activity. And we're talking about the stereochemistry, we're talking about shape, polarity, all that other kind of stuff. Understanding the relationship between the structure and the way it works is called SAR, structure-activity relationship. Now, you can actually do this mathematically to get QSAR, which is quantitative SAR. There's equations for doing that. All right, I think at this point we'll quit. We'll talk a little bit more about some of this stuff on Friday, but not all day. So we'll pick up with our case studies on Friday. See you then.